Neuromuscular junction. The amount of reactions and steps that occur on a molecular level when our muscles contract are fascinating. This video here is going to give us an idea of what happens during muscle contraction at the neuromuscular junction. An easy way to go over this is to kind of group them into 4 different stages. Here we'll have them grouped as number 1, excitation, number 2, excitation, contraction, coupling, number 3, contraction, and number 4, relaxation. This model shown here is a great way to explain muscle contraction with the neuron at the top of the model and the muscle fiber at the bottom. Let's zoom in to the top portion, which represents the neuron. First, I will clarify what some of this stuff is here. If you look outside of the neuron, you'll see towards the top of the picture there are these buttons that are black. Those represent calcium cations. The lightning bolts that you see present represent what we call action potential. The little red tubes that seem to go from outside of the neuron to inside are called calcium voltage gated channels, or calcium VG channels. We will also see several vesicles with these little green things that says ACH, and that stands for acetylcholine. Action potential is an electrical signal or impulse that is generated and propagated along the membrane of a nerve cell. It can be thought of as a rapid sequence of changes in the voltage across the membrane. Concentration gradients of ions can cause action potential. In the example here, the action potential opens the voltage-gated calcium channels. This allows the calcium ions into the axon terminal from outside the cell. Inside the axon terminal, we can see vesicles filled with the neurotransmitter acetylcholine. The calcium ions having a positive charge will be attracted to the negatively charged vesicle. These vesicles are called synaptic vesicles. The calcium binds to a membrane receptor protein on the vesicle called synaptotagmins, and there are areas on these receptor proteins called calcium binding loops. They're sequences of amino acids with negative charges on them. The positively charged calcium ions are drawn to these negatively charged regions. When calcium binds to these proteins, it triggers a response for the vesicle to release the neurotransmitter acetylcholine into the synaptic cleft. That's basically the space between the neuron and the muscle fiber. This can also be referred to as the synaptic space. Now we start to transition to the next step, which is excitation-contraction coupling. The acetylcholine goes from out of the axon terminal into the muscle fiber to a receptor called the cholinergic receptor. These receptors can also be called ligand-gated sodium ion channels. These receptors are located at the superficial portion of the sarcolemma, which is the plasma membrane for the muscle fiber. Notice that there's some sodium ions floating in the synaptic cleft up there. You'll see in this image that the sodium ions are no longer in the synaptic cleft. And what basically has happened is when the acetylcholine binds to the cholinergic receptors, the sodium channels open, allowing sodium ions to enter the cell and generating in their own action potential along the sarcolemma. An easy way to understand it is they have a positive charge, and when they go into an area with a negative charge, they bring their charge with them, thus changing the charge of the region. In this case, it would be the muscle fiber. The T tubules you see are also known as transverse tubules. As you can see, are really close, and they kind of go inside the sarcoplasmic reticulum. This influx of sodium ions forms its own other active potential. The action potential generated from the influx of sodium ions causes voltage-sensitive proteins located in the T tubules to change their shape. The official name for these proteins are called dihydropyridine receptors. These proteins are also connected to another set of proteins that are part of the sarcoplasmic reticulum, which are called ryanodine receptors. And so these two proteins are important because they pretty much connect both the T tubules and the sarcoplasmic reticulum together. When the dihydropyridine receptors change shape, it triggers the ryanodine receptors to open, which releases the stored calcium ions out of the sarcoplasmic reticulum, where they're being stored into the cytoplasm of the muscle cell. We're now starting step 3, regulatory proteins and contractions. What you'll see here is the calcium ions have left the sarcoplasmic reticulum. They go down the sarcoplasm, which is basically the cytosol of the muscle fiber, and then the calcium ions bind to what is known as troponin in this photo. At the lower left hand of this corner, you'll see a blue arrow and a purple arrow. The blue arrow is pointing to the troponin, and the purple arrow is pointing to what is the tropomyosin. Both the troponin and the tropomyosin are a complex of proteins, and they work as regulatory proteins. Below that, you'll see this little blue thing, and that's basically the actin, or the thin filament, and the tropomyosin is covering it. What the calcium ions are doing is they're binding to the troponin, which then changes the shape of the protein. When the shape of the troponin is altered, it pulls the tropomyosin away from the myosin binding sites. So what happens next is the troponin shape changes, and this creates a displacement of the tropomyosin. This leaves the actin binding sites exposed, and the actin binding sites are the parts of the thin filament that the thick filament will grab onto. The spider-looking thing in this image is actually the thick filament, whereas those little blue things that are covered by the troponin and tropomyosin complex is the thin filament. Over here, you'll see in this image a little circle in green, and that's just basically my way of showing where the myosin heads have grabbed onto the active sites of the actin. The yellow arrow is what the myosin is, and that's the thick filament. What happens when this contraction occurs is the sarcomere shortens, and the proteins of both the myosin and the actin, the thick and thin filaments, overlap more. So the actual proteins themselves don't decrease, but the sarcomere space becomes, smaller as it contracts because there's a larger overlap. When the myosin heads of the thick filament attach to these spaces, this process is called cross-bridge cycle, and ATP will be used to help facilitate the cycle by providing the energy needed for deattachment, reattachment, and pivoting of the myosin heads. What we can take from all this is, as long as the calcium ions are present in the sarcoplasm and they bind to the troponin, it will keep the actin binding sites unshielded. Also, as long as ATP is available to facilitate the cross-bridge cycling and the pulling of actin strands by myosin, the muscle fiber will continue to shorten until it reaches its maximum limit. The fourth step we have here is going to be relaxation. When the nerve stimulation stops, so does the release of the acetylcholine. The acetylcholine unbinds from the receptors of the muscle fiber. This is where an enzyme comes into play. Acetylcholinesterase breaks down the acetylcholine in the synaptic cleft so it does not rebind. Therefore, the molecules of acetylcholine cannot be reused. The little Pac-Man looking character at the top of the picture is the acetylcholinesterase, the enzyme. In this image, you'll see a couple different things. So we see that the enzyme acetylcholinesterase has pretty much broken down all the acetylcholine. And you'll see a couple different things. You'll see some purple arrows, and that will just kind of indicate that the thick and thin filaments have gone back to their original states, and that's because the troponin and the tropomyosin complex are both now over the active sites of the actin, so the myosin heads can no longer bind to it. What you'll also see is these blue arrows that point to the fact that the calcium is now back in the sarcoplasmic reticulum. This occurs because of an enzyme called sarcoendoplasmic reticulum calcium ATPase, or SERCA for short. We'll just keep it that way. Now, what do we do about all this sodium in the T-tubules that have entered the cell? Well, we have a thing called a sodium-potassium pump, and it's going to help also not only pump the sodium out, but it's also going to bring back the charge and depolarize it, which means basically bring it back to where it was initially. What this will do at this point is return the energy gradients to their normal state. So this is not my best work, but this is my picture of a myofibril. And within a myofibril are these things called sarcomeres. They start at both Z discs here. So you see the little hand on the screen. This Z disc right here is pretty much made a protein called connectin, and it anchors all the thin filaments, also known as the actin. So that would make this whole thing right here the thin filament. Over here you have the M line, and that's very similar to the Z disc, but what it is it pretty much anchors the myosin or the thick filaments. And so that's pretty much that for that. For the H zone, this is a very interesting section here. So this is the part of the myosin where there is no overlap from the actin, because as you see, some of this overlap is right here. Now, the A band is the whole area of myosin. It's usually darker when you look at it under a microscope, and it's because of the overlapping of proteins. You'll see normally in the microscope how a muscle cell is striated, which means striped, and it's caused by these overlaps. The I band, which is the next description here, that is basically where just the actin is present, where there's no overlap from the thick filament. And the reason why it's lighter in color is since they're thinner, there's more light can go through in a sense. So it's basically the overlapping of proteins that makes it appear to be striped. And you see right here, this is where one sarcomere starts, this is where the next one does, and this would be another sarcomere. And again, this is another example of more actin, or the thin filament. We see more up here just to kind of give you an idea, and then here we see the thick filament again. And we see the same process here with the M line, and both here and here.